So to use your terminology, and tie this to say the PTSD research with MDMA, the person with PTSD has this traumatic memory where the episodic memory, the thing that they're remembering, is tied to this very negative emotional state, but the engram is in this closed state, and those two things are locked together. And MDMA in this case is putting it into the open state, which won't dissociate those two things on its own, but it will put you in this metaplastic state where, in this case, the psychotherapy then can decouple the negative affective component from the actual memory.
Perfect. Exactly right.
So let's get back to the autism stuff. So autism's interesting with respect to MDMA. So even though you've told us everything about, MDMA, the acute effects might not be the thing that has to do with this critical period reopening. Autism is interesting with respect to MDMA in particular, because you can imagine that this sort of metaplasticity inducing effect is useful, but also for autism itself, as opposed to other conditions, the acute effects seem like they would be quite useful as well. And so is anyone looking actively right now at human clinical trials for MDMA for autism?
Yeah. So there has been one study that was led by MAPS, and Bera Yazar-Klozinski is the first author on it, that has looked at MDMA's acute prosocial effects and its ability to attenuate social anxiety phenotypes in autism. And so what they found but I just want to clarify that, autism, even though people, think of it as a disease of the social brain, and certainly there are subtypes of autism where social anxiety is a big part of the disease. Social anxiety is not actually a central core symptom of autism. It's seen in some causes, but not others, right? The core feature of autism, social impairments, really center around this social cognition, which, I draw the distinction because I think that MDMA, what it's doing in the acute setting is it's triggering, widespread oxytocin release, probably from those magnocellular oxytocin neurons, as well as the parvocellulars. And that social anxiety component of it seems to be the magnocellular neurons. Those neurons send projections to the amygdala, and they really seem to, that social anxiety part of it seems to be mostly around the amygdala. But but they were not able to, with the acute sort of effects of MDMA, they were not able to correct the sort of classical autism symptoms, the sort of social cognition part of the disease, right? And so it was a help, but it wasn't for that. And so that's interesting, and I'm glad that study got done, but and it will probably be useful to some people, but it's different from the way that we are thinking about how we might use MDMA to help people with autism. And I have to say, we have to be a little bit careful here because, one of the things that we found when I was a graduate student with autism is that their ocular dominance plasticity phenotype in the FMR1 knockout mice that we were studying as a model for autism, they seem to have a little bit of hyperplasticity. So their critical periods don't close as fully, and they're able to sort of induce a little bit more plasticity than normal, even when they're juveniles. And so, I think we're gonna want to really test this a lot in human patients, in animal models, before we jump to the idea that even in the first scenario I described, where we would sort of, pair the biochemical intervention with their critical period reopening, before I would recommend that we jump into clinical trials to do that, I would want to make sure that we're not going to have some other impact on critical period reopening in general, now that we think that, the psychedelics might be this sort of master key. So I think I want to be a little careful there.
And so given that multiple psychedelics have this effect, given some of the mechanistic commonalities that some of these drugs have and the evolutionary conservation of some of these receptors, when you think about just like the core molecular basis for metaplasticity. Is it the serotonin system that's specifically involved here? How much work has been done at the very mechanistic level in terms of metaplasticity generally?
Yeah. So basically, we literally just submitted this paper on Monday. I'm not really able to talk about, some of the results until they're published. But what I can sort of tell you is that, the dominant theory in the field is that, it's sort of focused on this idea that there are serotonin psychedelics and non-serotonin psychedelics. And there's been some really beautiful work from Brian Roth's lab showing the crystal structure of LSD bind to the serotonin 2A receptor, and it just sits in there and it activates it for a really long time and triggers a different mode of biochemical signaling through beta arrestin. And I think it's got a lot of people excited about the idea that serotonin 2A beta arrestin combo sort of explains both the psychedelic and the therapeutic effects of psychedelics. But what we've been working on is we have some evidence now that it's not that, right? That while LSD and psilocybin require the 2A receptor, the rest of the psychedelics that are able to reopen this critical period can do it even if we block the serotonin 2A receptor, even if we genetically engineer the beta arrestin knockouts, right? Like even under those circumstances, we're able to do it. So while it seems to be a mechanism, it is not the universal mechanism.
What's an example of that where you reopen something after blocking 2A or preventing that pathway from coming on?
I mean, without getting into too many details, in this paper that we just submitted, we looked at a bunch of different psychedelics.
I see.
All of them reopen the critical period, and some of them, but not others, require the 2A receptor, right?
Okay.
Some of them, but not others, require the beta arrestin mechanism. And so the goal of that paper really is to say, okay, if it's not, the universal mechanism is not at the level of receptors, if it's not at the level one downstream of that at biochemistry, what's the universal?
I see.
And we have some ideas, so stay tuned because we have some good ideas about what is the universal that all of them are able to trigger in order to reopen critical periods.
Are there any examples of these metaplasticity inducing drugs that we know of where there isn't a strong psychoactive component? And what are your thoughts on whether or not such a drug could be engineered?
Yeah, so I know that there's a big push, especially from the sort of people who are hoping to, come up with a marketable alternative to kind of engineer out the psychedelic, quote unquote, side effects of the psychedelic drugs. And again, in this paper that we've just submitted, we have some evidence that approach will fail, and that any attempt to remove or to shorten the time course of, the psychedelic activity will interfere with their ability to keep the critical period reopened. Furthermore, just sort of mechanistically, what I would suggest is that the what it feels like to be having this big psychedelic experience is actually what it feels like to reopen critical periods, right? So if you think about what people on psychedelics look like, right? It's like herding kittens, right? Like they're all over the place.
They often look like children.
Exactly. Then they will report, I felt like a childlike wonder for the world again, right? And they notice everything and they spend an hour like putting on a shoe and, whatever, like, and it's just like when children, like if you ever try to get children out the door on a snow day, oh my God, it's, impossible, right? They just cannot focus. And so even though habits get a terrible rep, that we're all habit-based, whatever, they make us efficient. They make it possible for us to get out the door. And so, this is probably why critical periods close. But, it's also, we think, just what it feels like to be in the open state. And so we think that if you try and engineer out that world is strange psychedelic experience, you're going to basically get rid of the thing that is making all of these plastic rearrangements possible.
I see. So your idea is really that the experiential component here is the effects, the psychedelic effects themselves are an experiential marker, perhaps, of the underlying molecular changes that are actually operative on the therapeutic side here.
Yeah, right. And I think that the reason that people have missed this in the past is that they've been overly focused on the non-context dependent markers like antidepressive effects or anxiolytic effects, right? Which we think are they are properties of these drugs, but they are not the properties that account for these sort of miraculous therapeutic effects, right? Like people who are quitting smoking after like a mega dose, not a microdose, a macro dose of psilocybin, they describe things like, smoking a cigarette just seems silly now because, I had this revelation that's the equivalent of like Santa Claus isn't real, and smoking a cigarette seems as silly now after my revelation as, leaving cookies and expecting presents the next morning. Like, it's just, people have had this like massive reappraisal, cognitive reappraisal. And I think that that's not something that is being captured by these attempts to model the antidepressive effects or the anxiolytic effects, because those things don't capture that context.
I see. Well, this is an interesting, I've had discussions with, everyone from David Nichols to David Olson, where, one says, I don't think you'll create these non-psychedelic psychedelics. And one says, I think they will. You're adding a very interesting twist to this, which is, it sounds like you would agree that, sure, we might make new drugs derived from psychedelics that are next generation anxiolytics, say, but they're actually not going to work for the things that do require these critical period plasticity modulations that you've been talking about.
Right. And I will tell you, we already have a little bit of evidence that that's true. So if you compare the clinical trials that were done for MDMA-assisted psychotherapy, right? So there, the model was very much in line with this sort of context dependence and the importance of focusing on, the psychedelic is the adjunct, but the therapy is actually the psychotherapy, the sort of getting at the root cause. And if you talk to the clinicians who carried out those trials, very much during the acute subjective effects of the MDMA, they were, making sure that the patient was, coming back to those traumatic memories, right? And there, in those clinical trials, and this is the Jenny Mitchell Nature Neuroscience paper, then you saw, big effects on PTSD, like cure. Yeah. I mean, just what we've never seen with SSRIs before, right? So like massive improvement over what's available now. Whereas in the psilocybin trial, this is the one led by Robin Carhart Harris, in that trial, they just kind of treated psilocybin as like, an SSRI, and they got basically the same
SSRI
effect.
Results. Yeah. Yeah.
They got SSRI results, right? Because they did not necessarily focus on that important context dependence, right? And so, I really think that, both David Olson and David Nichols are medicinal chemists, and they have a medicinal chemist's view of the brain, which is, we can put it in a dish. If we identify the receptor, we'll be able to identify the mechanism and we're done, right? But as a neuroscientist, I know that, memories, the things that make us behaviorally complex and rich, being able to speak, English in one context, and in my case, Turkish in another context, and not having any clue of how I'll ever learn Japanese, right? These richnesses of our behavioral repertoire are because of learned, experience-dependent, context-dependent capabilities of the brain that are not captured by just, neurons in a dish.